Something happened this week that David Sinclair described as the culmination of 30 years of work. Not a new paper, not another animal study. A real human being received the first ever dose of an epigenetic restoration drug, a therapy designed to reset the biological clock of living cells. The company is Life Biosciences. The drug is ER100. And for the first time in history, epigenetic reprogramming has crossed from the lab into a human clinical trial. Let us break down exactly what this is, why it matters, and what to realistically expect from here. To understand this milestone, you need to understand the underlying theory. For decades, the dominant view of aging has been that it is caused by damage, broken proteins, mutated DNA, worn-out cells. Sinclair's lab at Harvard has been developing a different idea, that aging is primarily an information problem. The epigenetic code, the system of chemical marks on your DNA that tells each cell which genes to activate, becomes corrupted over time. Cells slowly lose the instructions that keep them functioning properly. This loss of epigenetic information in Sinclair's framework is a root cause of aging itself. If that theory is correct, then the logical intervention is not to repair the damage piece by piece. It is to restore the information, to reset the epigenetic code back to a younger state. The tool for doing this is a set of 3 transcription factors called OCT4, SOX2, and KLF4, known as OSK. These are 3 of the original 4 Yamanaka factors, the proteins that can reprogram adult cells back into stem cells. That discovery won the Nobel Prize in 2006. The key insight from Sinclair's lab was that you could use a partial, controlled version of this reprogramming, expressing OSK without the 4th factor, as au be sûr, to restore cellular youthfulness without turning cells back into stem cells and causing uncontrolled growth. In animal studies, this approach reversed age-related vision loss in mice. It regenerated damaged optic nerve cells that were considered permanent losses, and it did so by resetting the epigenetic age of retinal cells to a younger profile, not by adding new cells, but by restoring the function of existing ones. Now, a human being has received this therapy for the first time. ER100 is the first clinical candidate from Life Biosciences' epigenetic restoration platform. It is being tested in a phase 1 trial for optic neuropathies, diseases of the optic nerve, including damage from glaucoma and related conditions. The trial is primarily evaluating safety and tolerability, with additional endpoints looking at changes in visual function. Sinclair, who co-founded Life Biosciences and serves as its chairman, posted this week that, quote, 30 years of research came down to one moment. That is a precise statement from someone who has spent most of his scientific career building toward this specific experiment. Now, the honest framing, because this matters. Phase 1 is not a proof of effectiveness. Phase 1 trials are designed to answer one question: is this safe in humans? We will not know whether ER100 actually improves vision or resets epigenetic age in patients until later trial stages. The full path from first patient to approved therapy, if everything goes well, typically takes years and multiple phases. Many drugs that clear phase 1 still fail in phase 2 or 3. That is the reality of drug development. What makes this significant is not what we know yet. It is what we are about to find out. Epigenetic reprogramming has been studied in cells, in organoids, in mice. The biology is compelling, but all of it has been preclinical, meaning no humans. Now, for the first time, we will have human data. If ER100 is safe in phase 1, and if even a modest signal appears in the visual function endpoints, the scientific community will have its first evidence that partial OSK reprogramming can do in people what it did in animals. That result, if it comes, changes the conversation not just for eye disease, but for every age-related condition that Life Biosciences plans to target with this platform going forward. The realistic timeline here is years, not months. This is phase 1. The finish line is a long way away. But what crossed a threshold this week is the scientific bet itself. The hypothesis that you can reset the epigenetic age of damaged tissue, not by repairing it one broken protein at a time, but by restoring the information that tells cells how to repair themselves, is now being tested in a human being for the first time. Watch for the phase 1 readout, not for efficacy data. We will not have that yet. Watch for whether the biological markers move in the direction the animal data predicted. That will be the first real-world test of whether 30 years of epigenetic aging research points somewhere that actually works in people. That is this week's breakdown. A 30-year scientific hypothesis just entered its first human trial. The field is watching closely, and so are we. Subscribe to BioHackers Longevity for weekly breakdowns of the most important developments in longevity and aging science. And if you found this useful, drop a comment below. We read everyone. This information is for educational purposes only. Always consult your doctor before making any changes to your diet, supplements, or medication.